Today we're going to be learning about the molecules that are important in cellular structures, and this information is in Chapter 2 of your textbook if you need to look up anything that we discuss here. There is a corresponding Power Notes sheet for today. It says at the top, Organic Chemistry and Functional Groups Power Notes, and you'll see that it has a few charts to fill in, especially on the back, and some diagrams here. So these, especially at the end, these diagrams, having that available will be really helpful for you as you are learning about functional groups. So do take some time to make sure that you have this document downloaded and or printed. So let's go ahead and talk about these molecules. And to begin our discussion, we need to define a couple of terms, organic versus inorganic. Now, these are terms that you've probably heard in other contexts before. When we talk about these terms in biology, we are talking about inorganic substances as being molecules that do not have carbon to carbon or carbon to hydrogen bonds. This is typically going to be your non-living material. And examples of inorganic substances would be the oxygen in the air that we breathe. Metals would be inorganic. Rocks, water, H2O. Water has no carbon in it whatsoever. So since it doesn't have carbon, it can't have carbon to carbon or carbon to hydrogen bonds in it. So that would make it inorganic. Organic substances are what we focus on in the study of biology because living things are mostly made out of organic molecules. These are substances that do have carbon bonded to other carbon or carbon bonded to hydrogen somewhere in that molecule. So essentially, since it has to have either a carbon-carbon or a carbon-hydrogen bond, organic molecules by definition always must contain carbon. They will usually also contain things like hydrogen and maybe some other elements, but they always must contain carbon. These are predominantly going to be the molecules from living things. It's either in a currently living organism, or it could be a once living organism that's now dead. It still counts as an organic molecule. So examples would be wood, that's just from a tree, right? Grass. Diamonds are all carbon-carbon bonds, and they come from pressurized remains of organisms. And petroleum, natural gas, when we process that becomes gasoline. That is a hydrocarbon, meaning it has a lot of hydrogen and carbon in it. So when we talk about other elements that are found in living things, I've already mentioned hydrogen. Hydrogen is a really small atom. As an element, it only has one proton, and that means it only has one electron, and so it forms a single bond when it's in a molecule. Chemists use a specific color coding scheme, and hydrogen is normally color-coded white in this color scheme. Oxygen forms two bonds, and a lot of times those bonds will be in the form of a double bond. Oxygen double bonded to another oxygen is the oxygen in the air we breathe. That's what O2 looks like. So oxygen always forms two bonds, and the chemical coding color scheme for oxygen is red. Nitrogen forms three bonds. So every time you see nitrogen in a molecule, it needs to have three bonds coming out of it. And its color in the chemical color scheme is blue. And carbon forms four bonds, and the color coding for that element is black. Sometimes gray instead of black is what you see, but normally it's one of those two colors. So in this picture of a molecule here, without any elements listed at all, we can tell that there are 1, 2, 3, 4, 5, 6, 7, 8 carbons just by the color coding. These two red are the oxygens, here's a nitrogen, and then all of these other 1, 2, 3, 4, 5, 6, 7, 8, 9 atoms are hydrogen. So we can tell that through that standard color scheme there. And a really helpful mnemonic device for remembering this would be 1, 2, 3, 4 honk. So if you can remember that, 1 bond for hydrogen, 2 bonds for oxygen, 3 bonds for nitrogen, and 4 bonds for carbon. So we need to make sure that we all are on the same page in terms of how molecules are drawn. And I'm mostly going over this because you are going to see molecules drawn in a variety of ways. So these 3 pictures at the bottom of the slide here are all showing you glucose, but they are highlighting different aspects of that molecule. In this first diagram, we have what's called a ball and stick model. If you were to build a 3D model of glucose, it would look something like this. And this highlights the color coding. You can see the carbon here is that dark shaded gray to black. You can see the red is the oxygen, and then the white circles are all hydrogen. You could also draw that in the center. We have what's called a Lewis structure. If you are highlighting the bonding and that's what you are concerned about, you might want to draw a Lewis structure. Every single atom is given its abbreviation like it would be on the periodic table, and you see lines to indicate the bonds in between them. Now, if you look at this third drawing, as you look at it, it might not look like the same molecule as what we have here. This is how a chemist would draw it. This is kind of the abbreviation for a Lewis structure, and it's called a line drawing. And the thing about it is that chemists understand that every time you have these 2 lines meeting in some kind of intersection, or if there's like a corner right here between the hexagon at the bottom and the OH over here, every time you have a corner, there's a carbon. So notice how those carbons are actually written in over here with C's, but they are not written in over here. That's because when a chemist is studying these molecules, it gets really tiring writing C every single time there's a carbon, because carbon is found in so many molecules. So instead of writing a C every single time a carbon is found, chemists know that this shorthand here, if there's nothing written, it's implied that that's a carbon. Also, notice how this carbon right here only has 3 bonds coming out of it. Well, we know from 1, 2, 3, 4 honk that carbon actually needs to form 4 bonds. So a chemist would recognize this and know that carbon forms 4 bonds and understand that that 4th bond would be filled with a hydrogen, like you see over here. This carbon has a hydrogen, and that's not given over here just as a shorthand or an abbreviation for a chemist. So chemists understand how to interpret that drawing. So if you see drawings like this and there are no carbons written on it at all, just understand that at every single corner there is an implied carbon atom. And all of those carbons have to have 4 bonds. So if 4 bonds aren't shown, you know that whatever isn't shown has to be filled by a hydrogen. Now, the reason why I'm going through this is not to have you draw these molecules from memory, but because you will see lots of pictures of lots of different molecules, and I want you to understand what you're looking at when you see that. So now let's talk about how these molecules are built. When you build large molecules, those large molecules are made out of subunits called monomers. Mono, M-O-N-O, means one. Mer, M-E-R, means a part of something or a section of something. So a monomer is one small part. Carbon atoms join together to make what we call a carbon backbone or a carbon skeleton. So if you have one carbon bonded to another, that would be the subunit to make a larger molecule. Polymers, on the other hand, are the large molecules that are formed as the result of linking together monomers. So poly means many. So polymer has many parts, many repeating subunits that are all covalently bonded together. When we talk about macromolecules, which will be the next topic of discussion in this unit, macromolecules are essentially gigantic biological polymers. And those would be things like carbohydrates, proteins, lipids, nucleic acids. These are all really big molecules, and they're made out of one subunit linked to another, linked to another, linked to another. And so we need to talk about how that linking happens before we can get into the specifics of what those macromolecules entail. So the process of linking monomers together is called dehydration synthesis. It's called dehydration because a molecule of water is removed during the process, and the synthesis part comes from the fact that you're building a molecule. Since you're building a molecule, this is going to require the input of energy. It takes energy to build things. If you think about building a shed in your backyard, if you have a pile of wood, it's not just going to fall into the shape of a shed. You have to input energy to pick it up and to fasten it together and make that structure. So what we have here is a diagram showing dehydration synthesis. Notice that we have on the left two monomers. This is a glucose molecule, and then we have a plus sign, a second glucose molecule. And we're highlighting these particular parts here of that glucose molecule because in order to join these two glucoses together and make this double sugar here, what happens is those two hydrogens and the one oxygen are going to come together and form a water molecule. Now you remember from 1, 2, 3, 4 honk that leftover oxygen, the one that used to be bonded to that hydrogen right here, when this hydrogen leaves, that oxygen is going to have to form another bond. So what happens is that bond forms right here, and now we have a covalent bond joining those two monomers together. So we have created a larger molecule out of two smaller monomers through the process of removing a water molecule, which is called dehydration synthesis. Hydrolysis is going to be the opposite of that. Hydrolysis is used to digest things or break down molecules. So in order to break down a molecule, you will have to add a water molecule to break that bond. And the word hydrolysis hopefully will remind you that that's what's happening. Hydro means water, lysis means splitting. You add water to split a molecule. And this is going to release energy. When you break down molecules, that is an energy releasing process. That's why we digest molecules to get energy out of the food that we eat. So here we're starting on the left with that molecule that we just built, right? See how they are two monomers linked together with that oxygen in the middle. Now this is hydrolysis, so we have to add water. So here is our water molecule that we are adding. So what happens to that water molecule? Well, it's going to essentially attack that bond, and the water molecule, an OH, is going to go onto one monomer, and the other hydrogen goes onto the other monomer when they get split apart. And so now we have added water, we have broken a bond between two monomers, and now we have two individual molecules, whereas at the beginning we had one larger molecule. So here's the two molecules that we have at the end. So this concept, hydrolysis and dehydration synthesis, both together, are super important in understanding how we use molecules, how we build them, and how we break them down in our bodies. So if that did not make sense to you, I urge you to stop and go back and replay that section in this video so that you can really understand what we just discussed. The next part of your notes is going to be focusing on specific functional groups. So at this point, you should be on the back of the Power Notes sheet where you have this chart to fill in. And in the chart, you are drawing or describing the functional group, you're giving its chemical formula, you're stating whether it's hydrophilic or hydrophobic. We'll talk about those terms in just a minute, and then giving some examples. I cannot stress to you enough how important it is for your practice and for your study to actually put the information in the chart and practice drawing and reinforcing what each functional group actually entails. So definitely make sure that you are keeping up with this either on your Power Notes sheet, or if you have a separate sheet of paper where you can draw these, that will help you immensely. So to begin with, let's define what is a functional group anyway. Basically, a functional group is just a group of atoms that when they are all bonded together, they have specific chemical properties. And these chemical properties are going to dictate how a molecule is going to react inside of a cell. This will dictate how the molecule participates in different chemical reactions. So it's important to be able to recognize them so that you can understand chemical properties of a molecule. We need to discuss the terms hydrophilic, which means literally water-loving. So hydrophilic molecules are soluble in water. They mix with water very easily, versus hydrophobic. Hydrophobic literally means water-fearing. Those would be the molecules that are not soluble in water. In other words, those would be the nonpolar molecules because they don't mix with water. So to begin with, we're going to start with probably the most important functional group for you to recognize because you will see it so often, and that is the hydroxyl group. So you'll notice at the top, I have a drawing here, and it is color-coded. Red, remember, is oxygen, and white, that's a shaded white, even though it kind of looks gray here. That would be hydrogen. And in all of these diagrams, you're going to see something called R. R is just a variable. Like in algebra, X could represent any number. R is going to represent whatever the rest of the molecule is. So there's some larger portion of a molecule attached to this OH group, and instead of drawing out the whole molecule, we're just using R as an abbreviation. So a hydroxyl group is just one hydrogen paired with one oxygen. And when I say paired, I do mean covalently bonded. That's a covalent bond. We remember that oxygen is very electronegative, so that's an unequal sharing of electrons, so it would be a polar covalent bond. The symbol is just OH. And this does not mean negative OH. This line here, it's longer than what a negative sign would be. This just means the OH is bonded to, that represents a bond, some portion of a molecule. Remember, we're just looking at one group of atoms that are a part of a molecule. So that's why we're showing that this group of OH is bonded to something else. I could put an R in front like there is in this diagram here. It would mean the same thing. So we just talked about the polarity of that covalent bond. So it is important for you to know that hydroxyl groups are polar, meaning they do form hydrogen bonds with other polar molecules such as water. That makes them soluble in water or hydrophilic. And the type of molecule that you will see hydroxyl groups on would be mostly alcohols and sugars. Those are the most important molecules that you'll see with hydroxyls. Hydroxyls are really common on a lot of different kinds of molecules, but they are kind of a defining feature of alcohols and sugars. Next, we have the carboxyl group. So in this diagram here, remember black is carbon, we have two red, those are two oxygens, and then the white is hydrogen. Notice there's a double bond here. There's two lines connecting this carbon and oxygen. That represents a double bond. So your description of a carboxyl group is literally a carbon atom that's double bonded to an oxygen, but then there's a single bond to an OH. Now, because this OH is not just a standalone functional group, because it's a part of this larger entity here, that if you were to notice this on a diagram and try to identify the functional group, don't call this a hydroxyl because it's part of something larger. So you would identify the larger functional group, the more inclusive group, as a carboxyl group rather than saying this is a hydroxyl. Since it's part of something bigger, we would identify that as carboxyl. The symbol that you see for this, it's normally written COOH. And the O's are written twice because they're bonded differently, and so a chemist would note that by writing the O twice. Instead of CO2H, you don't really see that. You see it written as COOH. This is also hydrophilic. It's got an OH bond there. Oxygen is very electronegative. That's going to make it partially negative. So it will attract other polar molecules like water. And this also is going to be an acidic functional group. Sometimes it's actually called carboxylic acid. Remember from your water notes, acids release hydrogen ions in solution. So if you were to dissolve a molecule with a carboxyl group, this hydrogen would leave and start to float around in the solution, and that's what makes it acidic. Your examples here, it's really common in a lot of biological molecules, but especially the two most important ones to recognize would be amino acids. The acid part of the amino acid comes from the fact that carboxyl is acidic. Also, fatty acids have a carboxyl group. And again, fatty acid, the acid part comes from the fact that the carboxyl group is acidic. Now we have our amine group. The amine group is going to be a nitrogen, which you can see in blue, bonded to two hydrogen atoms. Now notice in this diagram, it has a third hydrogen. This H plus is joining in, and that's because this group actually attracts hydrogen ions from a solution. And hopefully that sounds familiar to you from our discussion about acids and bases. We'll discuss that further in just a minute. Your symbol for the amine group is simply NH2. So the amine group is this nitrogen and two hydrogens before the hydrogen joins in. This H plus is an ion that is being attracted to the nitrogen, but it's not initially part of the group, which is NH2. Nitrogen, like oxygen, is also electronegative. So this is a polar functional group. It can form hydrogen bonds, soluble in water. It's hydrophilic. All of those are true for the amine group. And the amine group is actually a base. Remember when we talked about acids and bases? Acids give off hydrogen ions, bases absorb them. So in this picture here, what you're seeing is the initial molecule absorbing that extra hydrogen. So this is illustrating the fact that an amine group will absorb H plus ions from a solution, and that is our definition of a base. If you need to review that again, go back and look over the properties of water notes where we discussed pH. You find amine groups in molecules such as caffeine, but probably most famously, you find them in amino acids. That's where the amino part of an amino acid comes in. So an amino acid always has an amine group, and it also has that carboxyl group, which gives it the acid part. Next up, we have a phosphate group. A phosphate group is probably the easiest group to recognize because it's the only one with phosphorus in it, and it has phospho in the name. So a phosphate group is phosphorus bonded to 4 oxygen atoms. One of those oxygens is double bonded, but the rest of them are single bonded. The symbol for phosphate is PO4 2-. That 2 means it has a -2 charge. You can actually see that up here. These two oxygens have lost their hydrogens, so that gives it a -2 charge. Since it is losing those hydrogen ions, that also makes phosphate acidic. Remember, acids lose hydrogen ions when you put them in water, so that makes phosphate an acidic functional group. And those negative charges also make it very attracted to water. So it is also hydrophilic, even though it does not say hydrophilic on the screen, it does dissolve easily in water. So where do you find phosphate groups? Probably most famously in DNA and RNA. There's lots of phosphate in those molecules. Phospholipids, it's even part of the name. And then ATP, that energy molecule of the cell. NADP, I know you don't know what that is yet. We will discuss that in our next unit, which is about metabolism. And our final group is the methyl group. A methyl group is kind of the outlier here. It's a carbon atom bonded to 3 hydrogens. So it's just carbon and hydrogen. That should start to sound familiar to you. CH3, just carbon and hydrogen, no oxygen, no nitrogen, no nothing else. That means CH bonds being nonpolar, the methyl group is going to be your only functional group that is hydrophobic, meaning it is water-fearing. It does not dissolve in water. It does not mix with water very well or at all. So the methyl group with its CH bonds is going to be hydrophobic. Methyl groups are really interesting because what they do in your body, they can actually control how a molecule functions. Because it's hydrophobic, it will make a molecule react a different way. And so this is one of the common ways to turn genes on and off. You have genes in your DNA that are expressed, and so we can see the physical expression of that, but some of them are turned off and not currently being expressed. And methyl groups play an important role in that. They also play a really important role in sex hormones like estrogen, testosterone. There's a lot of methyl groups on those molecules. So to leave you here today, I have a picture of 6 different molecules here that I want you to look at, and I want you to try to pick out the different functional groups from these molecules. So on your Power Notes sheet, underneath this chart, you also have this picture available to you. So these, it looks like these are just 5 of the molecules. It looks like I added a 6th on the PowerPoint. So you can certainly look at all 6 of those molecules and then just look through and try to pick out what functional group you see on each one. I recommend maybe color coding it and then maybe write down OH is red and then, or hydroxyl is red, and then you would circle anything that has a hydroxyl group on any of those molecules. Go ahead and do that as a way to practice this material, and we will go over that in class. So hopefully you found this helpful. Please go back and rewatch any part of this video that you did not understand, and if you have questions, bring those to class with you so that we can discuss them.